Thank you. Thank you. Good afternoon, all of you. We are here today for the third live session. So people are yet to join. However, I can see one participant, Priyanka. So Priyanka, can you hear me?
Yes, sir. Sir, good afternoon.
Good afternoon. Yeah. So Priyanka, we will wait for some more people to join.
Okay, sir.
Yeah, yeah. So you have attended the earlier two live sessions as well, Priyanka?
Yes, sir. Yes, sir.
Nice, nice. Okay. So do you have any specific queries by the time people join? Maybe if you have any queries, I can work on that because this is an opportunity. You are the only one here right now. Or you can think and let me know if you have some queries.
Sir, I have queries.
Right.
So, can I ask now?
Yes, yes, sure. And if you can refer to me the particular lecture, that will be a little bit helpful. Anyway, you first ask me. Yeah.
Sir, I want to ask about CRISPR. Why CRISPR gene drives are called selfish genetic elements?
Okay. I think it's a very interesting question, although it was covered in the last module lecture. Particularly, you can see they can sometimes self-drive. So we discussed somewhere. I mean, you can see their asymmetric inheritance sometimes. So that's why these kind of gene drives are selfish, because they only know about their own survival and own proliferation. Is that simplified for you?
Yes, sir. Sir, okay, I got it.
Yeah, yeah.
Sir, I
So
have other
generally,
questions also.
Mendelian genetics, the inheritance is 50-50. Say
Yes, sir.
for any two, I mean alleles, right? But in this case, only one will dominate because that tries to take the upper hand and it skews the inheritance in its favor. So they act like selfish people. They are only protecting their own interests, right? So that's why we call them as selfish genes, or these drives are selfish.
Okay, sir.
Yeah, yeah, right. Any other query you may think and let me know.
Sir, you've told CRISPR in bacteria. So is CRISPR an efficient and successful technique in viruses also?
No, CRISPR is a tool which is universal. I mean, you can apply it to editing almost all kinds of genomes, whether it is viral genome or whether it is bacterial genome or mammalian genome. But if you're asking about CRISPR as a basic protection mechanism, it is not available in other organisms other than the bacteria. Which part you are asking basically?
Sir, I want to know about it, whether it can be used as an efficient tool in case of viruses.
For editing viral genome?
Yes, sir.
Yes, it can be used to edit viral genome as well, because it
Okay.
is a tool we use. But CRISPR exists as a immune defense mechanism in bacteria, right?
Yes, sir.
Are you interested in that part? No.
Sir, I've studied.
Okay. So
But it was, I mean, there was some gap. It was seventh week lecture.
right. So your simple question is whether CRISPR Cas9 can be used as a genome editing tool for editing viral genomes. Am I correct?
Yes, sir.
Yeah, it is. You can edit it.
Sir, it is as efficient as in case of viruses. Oh, sorry, bacteria.
Yeah, any genome you
Okay.
take, any genetic sequence you take from any source, and you use this CRISPR-Cas9 system, it will edit it with equal efficiency.
Okay.
Fine.
Sir, so can we use micropropagation for delivery of CRISPR gene therapy in viral cultures?
Micropropagation for CRISPR.
Gene therapy delivery?
Micropropagation is something which is associated with tissue culture.
Okay.
Right. So I think you are mixing up things here.
Okay.
Right.
Hmm.
So can you tell me once more the question? Reframe the question once more. Maybe I will
Sir,
also use of micropropagation for
delivery of CRISPR gene therapy in viral cultures.
I think no. I think two things are here totally different.
Okay.
Yeah. Micropropagation is big thing, and virus culture is something different. Virus you culture on live cells. Virus you cannot culture on simple media.
Yes,
Right?
sir. Yes, sir.
You
Yes,
need
sir.
virus cannot survive on its own, so you cannot cultivate it in the way you can cultivate culture bacteria
Okay.
or plant cells, plant tissue cells, as in the case of micropropagation or tissue culture. So first you have to grow the host cells, whether it is mammalian cell or insect cell or maybe other cell lines. Then you propagate the virus on these cell lines, right?
Yes,
Media
sir.
media you are using only for supporting those cell lines. So the cell lines survive and the virus is infected on these cell lines. So that is how you do the viral culture, right? Now, I don't think you can use micropropagation for delivery of CRISPR-Cas9 to virus cell lines, virus culture, right? These are entirely two different things.
Okay, sir.
Yeah. Please kindly read a little bit about these different techniques. So I have given me listed various methods of delivery of gene editing tools. So
Yes,
kindly
sir.
read, kindly go through all those gene editing tools which are already listed
Yes,
and
sir. Sir,
on which I have already given the lectures. Yeah.
sir, one more thing I want to ask. Will you please throw some light on designer viruses made using genome editing?
Yeah, it is although beyond the scope of this particular lecture, but yeah, since we have time, I can
So only short
discuss. Yeah, sure,
introduction.
sure, sure. Why not? Why not? So, see, what do you mean first by designer viruses?
So
Say, if you
genetically modified viruses.
genetically modified viruses, fine, but in what way designer?
For example, we can um, design to fight some kind of disease.
Right, I get it. So, let us go to the structure of a virus, a simple structure. Okay, so you have some nucleic acid, which is coated by some protein layer, right? Some capsid envelope, protein envelope. Okay.
Yes,
So
sir.
now, and then the virus has various genes inside, apart from these capsid coat protein genes. So it will also have certain critical genes which are important for its life cycle. Okay, so late stage, early stage, mid stage, late stage. So those are the various, in simple way, the various stages of a virus, and various genes act in these different phases of its life cycle. Now, some of the capsid proteins, uh, for example, in the case of COVID, you have a specialized protein which uh, helps in its pathogenicity. Okay, and there
Yes,
are
sir.
also other many such viruses where you have certain ligand proteins on the capsid, which play a role in the pathogenicity. So what you're telling is designer virus. So one of the things that you may think of designing is these kind of ligand proteins which are expressed on the surface of a virus. So your engineering principles may be used either to enhance the pathogenicity of these capsid proteins, code proteins, or ligand proteins, or decrease it, right? So one of the possible thing is that you may use a live virus as a potential vaccine candidate if you decrease the pathogenicity of its ligand virus by some kind of genetic engineering, right? So that can be one of the design principles. Or people may be using them for nefarious activities, like using them as biological weapons, and thereby, in that case, they may increase the pathogenicity by using the principles of genome editing, which is absolutely ethically wrong. So, which because you know the entire human race may be under danger, and these are the things we have dealt with in the ethics portion and the regulatory portions. What need to be regulated and what should be done which is ethically correct and beneficial for the humankind. So when going back to your simple question, whether we can use genome editing for making designer virus, the answer is single word, yes. So it clears your query, doubts?
Yes, sir.
Okay.
Sir, you told just reminded me of bioethics also. So in lecture 47, you have used the term regenerative medicine.
Yes.
Sir, regenerate can we use? We can use gene therapy as a regenerative medicine, right? So, will you please tell about some recent regenerative medicine in terms of gene therapy?
See, many of these things, as I have already discussed, are at various phases of development. Okay.
Yes,
And
sir.
some are under clinical trials. So the scope of regenerative medicine is basically much more focused on, say, the development of organs, say, for example, humanized organs in pig or some other animals. Particularly pig is considered because it is thought to have very close relation with the human species. So whatever gap is there in the genomic composition, we can narrow that gap by humanizing the pig genome, and thereby, and also targeting the special set of particular set of genes which are involved in the development and genesis of an particular organ. Say, for example, it may be liver, or it may be kidney, or it may be as simple as your skin. So if we use the pig organ directly, there will be rejection because xenografts are not well accepted. And then xenotransplantation has not been found to be that successful. And there are a lot of literature where people have tried experimenting with animal systems for xenotransplantation, and the success rate is not that high. And also to avoid immune rejection and graft rejection, a lot of anti-immune therapies has to be administered along with it. Now, when it comes to regenerative application of genome editing techniques in regenerative medicine, what do we target? we will look into the set of genes which are involved in the development of a particular organ, or the genetic network which is involved in the development of a particular organ. And with the use of bioinformatics, we'll try to find out the similarities and differences between the two gene sets and particular genes, candidate genes in question. Then, once we do that, we now know which are the alphabets in the pig, which we can edit out and change them to the type of alphabet which is there in the corresponding human gene candidate, right? So this way, we can use genome editing techniques for humanizing the pig genes and thereby produce organs which are more acceptable, more human-like organs in live pigs. So when we will be using these kind of humanized pig organs, the acceptability will be better than the xenografts. So this is the way overall, one of the ways by which genome editing has a lot of potential in regenerative medicine. So now you have another question to update you with a list of such achievements. So for that, I have to do some literature research, but whatever has been discussed in the class, that is some operating principles, which is of much importance. But as of now, I can tell there are no any humanoid organs available. But research is going on in that direction. So and also clinical trials once conducted successfully. So that is the future in which we may have organs from humanized pigs or some other.
Okay, sir.
Hello.
Hello, am I audible?
Yes, sir.
Priyanka, yeah, sorry. So I think the last point that I was discussing with you is that the organs from humanized pigs for regenerative medicine, and I think I could clarify your query.
Yes, sir. Yes, sir. I've got it.
Okay, thank you.
Sir,
So
I have one
yeah,
more question. Can I
sure,
ask?
sure, please go ahead.
Sir, in lecture 48, you have told that introduction of a new transgene into a wild fish population threatens the viability of both wild type and genetically modified organisms. Why is it so? why there is reduction in the viability of offspring.
Can you tell exactly what is the title of the lecture?
Lecture 48.
The title.
Okay.
This is genome engineered disease models. Which one exactly? Genome engineered disease models. Am I online, Priyanka?
Hello, sir.
Yeah. So, Priyanka, can you tell the title of the lecture? I mean,
sir,
which one?
Sir, meantime, can I ask question?
Yeah,
Hello?
let me do one thing. Yeah, sure, Rakesh, you can. So I can see two more people joining, Rakesh and Neela, right? Neela, am I audible to you?
Yes, sir.
Yeah. So let me actually tell, basically, we are here to discuss 4 modules today. Am I audible, all of you, please?
Yes, sir.
Yes, sir.
So we discussed up to module 8 in the last lecture, last live session. So today we are discussing about, we can have discussions on these 4 modules. Module number 9, which is genome engineered disease models, and then iPSC induced pluripotent stem cell models, then cancer disease models, and also engineered immune cells for cancer therapy, both lecture 1 and 2, and then also about personalized medicine and genome editing and personalized medicine. And the last part is the bioethics and the biosafety. So these are the overall modules we are here today. And in the meantime, Rakesh, you want to ask some questions, and Priyanka, if you can tell me exactly which lecture, so I can come back to you.
Sorry for the interruption, sir. It's week 12, first and second lecture PDF.
Week 12.
Oh, okay. Ethical concerns. Germline genetic.
Ethical concerns.
Yes, sir.
Lecture number 48, na? 48.
The page number 48 of the first
Bioethics,
PDF.
bioethics and biosafety, ethical concerns.
Yes, sir.
Here I have it in a different way.
48. Page number 48.
Sir, page number 48 of the first PDF.
What is the subheading? Subheading, can you tell? If you can share me in the chat box the particular slide, or you can write it down. Yeah.
Sir, is her subheading is risk and controversy surrounding the use of GMO. It is a lecture, sir, week number 12. PDF lecture notes are there, na? Lecture 1 and 2, week 12.
So it is number 2, regulatory issues in genome editing.
Week 12, sir, lecture 1 and 2. That PDF is there, na? In that, it is there, sir.
Page number.
Page number 48, she said. But heading will come under.
These are the same slides.
Controversy, yeah, same slide.
46 page number, sir, for heading.
Let me find out. I may have uploaded the risks and controversies. This one, can you see it?
Yeah.
Risks and controversies surrounding the use of GMOs, Priyanka. Hello. This slide, risks and controversies surrounding the use of GMOs.
Yes, sir. Sir, page number. It's on page number 48, about which I asked.
So this is the slide, risks and controversies surrounding the use of genetically modified organisms.
Yes, sir.
Yeah. Exactly what you want to ask here.
Sir, I want to ask. You have written that introduction of a new transgene into a wild fish population threatens the viability of both the wild type and genetically modified organisms. So why there is reduction in the viability of
Let me
offspring?
find out. I am unable to look at the.
Sir, page number 48. Sir, this one.
Okay. Right. Exactly what you want to do. What you want to understand here.
So why there is reduction in the viability of offspring after the introduction of transgene into wild fish population?
Reduction of the viability of the population having transgene, right?
Yes, sir.
Yeah, these are actually some of the kind of natural safeguards, So because transgene is not something which is existing in natural conditions. So eventually what happens, the transgenic population will go down because the pressure due to natural selection will be there on them. So that is one of the reasons the number goes down. So
Okay.
is it clear?
Yes, sir.
So is it fine?
Sir,
Yes, sir.
I have similar on page number 51, sir. Gmo and soil fertility. Same PDF. Same PPT.
Yeah. This one?
PPT. Yes, sir. No, later. This, yes, sir. So first sentence I am, sir, that GM crops transfer their genes to soil and bacteria. Sir.
Actually, this is little bit written in a simplified way. So there is a chance of escape because the reverse transformation takes place. So in the way we are transferring into the genetically modified crops. So here the reverse process may takes place, and then when it will happen, say we have taken some of these genes from microbial sources. So now they come back, and then some of them other microbes may carry these kind of transgenes. So that is some of these studies has been carried out by certain groups. So, and these are some of the concerns which has been raised due to these kind of techniques.
But generally, sir, it is people, scientists and other people say that they just, for example, if you are taking BT cotton and all other this BT related GM crops, whether this BT gene also got transferred into soil?
I don't have the specific information. So this particular study I will forward to you separately. So these are some of the concerns which are being raised.
Yes, sir, because this I am into plant biotechnology section area. So
these are some of the.
Yes, sir.
So, but many people, they are trying to find out the evidence which supports their concerns. So these are possibilities because in the way we are transferring genes into a system, the gene may also escape from the system using natural systems. Okay.
Yeah, because sir, this first sentence is making controversy, so that's why I was more on that things.
Right.
Because now,
yeah, you are correct. I also these are some of the concerns which are being raised in the regulatory bodies. I mean, some of the things which may happen if these kind of technology is not implemented after proper regulation. So that's why the biosafety concerns are being raised, and these are being implemented so as to make the technology much more safer. Is it fine, Rakesh?
Yeah, sure, sir.
Yeah. So Neela, can you hear me, Neela?
Yes, sir.
So if you have some queries, or Rakesh may have some more queries.
I'm not sure if this is a query, but I would like to know if these techniques such as talent, CRISPR, and the rest can be used in biofertilizers, methodology of biofertilizing. Is it possible?
Yeah, Yeah, I mean, your voice is a little bit disturbed. But anyway, I can get your some echo is there. You're asking whether these genome editing technologies can be used in biofertilizers.
Yes, sir.
Okay. See, biofertilizers means you are probably interested in fixation of nitrogen or something like that.
In more towards like since I'm learning about it, I was intrigued by it. Rather than only nitrogen fixation, others like inorganic materials such as phosphorus also
Right.
that can be easily brought back in for the plant's growth, rather than like in the
Yeah.
slide
So
of
okay.
PDF that it was
Yeah,
just
I
shown.
get it. Yeah.
Rather
So
than
yeah.
waiting for the whole session to reconcile or wait for the soil to bring back its fertility, we can use biofertilizer to enhance it. Is it possible?
Yeah, no, that would actually depend. See, this is a platform technology. Genome, all these techniques that you we have discussed here, TALEN, then Gelefan and CRISPR-Cas9. These are platform technologies. These can be used to edit genes and genomes, right? So now the question that you are asking is whether it can be used for applications in biofertilizer. So now probably here you are telling about some kind of enhancement, some nutritional enhancement in the final product, right? So those would be dependent on the expression of certain genes in those microbes which produces the biofertilizer. So it can be, say, your biofertilizer has enriched nitrogen content or phosphorus content. So that you have to map out what are the genetic pathways involved in that kind of a process, and accordingly you have a inherent capacity of the microbes already which can do that job. But if you want to increase the efficiency of the entire process and you feel some kind of introduction of new genes or some kind of genome editing can help in say optimization of the process, that you can use these technology to do that. So overall you have to find out what is your goal, what is the gene that you want to modify or edit or replace, which is going to enhance the efficiency of the process of biofertilizer production. So biofertilizer is not one thing, as I can understand. You have to target one by one. Say, for example, you only take 3 components NPK. So whatever genetic pathway is involved in any of these NPK, and you target, for example, the P or the K pathway or the N producing pathway. So you have to target the genes which will enhance the production of those, right? So you can definitely use it, but you have to find out exactly what is the scientific question that you want to ask or that you want to address. And in addressing that scientific question, what is the genetic change that you have to make and where, in which genes? So if you need some kind of editing, you can go for the editing, or if you need some kind of deletion, then you can do the deletion, right? So this is a platform technology. You can use it for any purpose which you can find out is controlled by genes, and by editing the genes you can either enhance the process or you can down regulate the process or even switch it off, right?
Yes, sir.
Yeah.
Okay. Thank you.
Let me find out if there are some other queries coming online. There are no any online queries.
Sir, my general question regarding.
Sure, please go ahead.
Sir, what about government of India's stance on this bioethics with regard to CRISPR-Cas technology? Whether any guidelines they are prepared other than that SD1, SD2?
Yeah, the Department of Biotechnology (DBT) is actually the concerned authority in this regard. So you can find out the latest guidelines and the policy papers in the CRISPR-Cas9. So I think in one of the lectures I gave you a full landscape. So that was in lecture number maybe Indian context. Not which lecture? Let me find out. Lecture number 12. Yeah, so I think these are some of the information already shared to you regarding the various, status of various diseases even. So, and you can read this lecture a little bit more and also visit the DBT website if there is any update on the recent developments. Because this lecture is prepared a little bit earlier. So in the last 10 months or 1 year, if there is some kind of a update, I also have to find it out because this was prepared long back and then recorded accordingly. So the latest one, if there is something, we have to find out. But for your exam purpose, we will go by what is being taught in these sites, right? So this is actually information we collected, some of the information collected from this particular paper. So this is in the proceedings, BMC proceedings, but the DBT website also have information which is updated from time to time.
Yes, sir.
Fine.
Yes, sir. Sir, regarding that, my previous question that whether I can get research paper, you said.
I will send some of those research papers who have expressed the concern. Yes, so what you do, you can send me your email. Yes, sir. My email is ubora @IITG, and probably it is given also somewhere. Let me check out. Yes, yeah, so this is available in my introductory slide. You can see my email here. So yes, kindly please send your email, then we will provide you the information. So wherever concern is expressed, and some of the hypotheses and some of the experiments where people have been trying to prove the concerns they have raised, right? Yes, okay. So is there any other question?
Sir, another question. Sir, CRISPR-Cas9 technology, actually we are editing the gene in itself there, right, sir? We are not taking foreign gene, right? So yes, this one can be treated as a conventional trading, plant breeding in context of plants, right? Plant breeding, we are doing mutation and then other crossbreeding things. So can we take as a mutation and this CRISPR as on same platform?
I think it's a very interesting question you are asking. So that is the reason why this technology is considered much more dangerous than your earlier genetic engineering technique. In genetic engineering, we always introduce, or maximum we do some kind of there also we can do some mutation. But here we can delete a gene entirely, and we can redesign, and we can cut the genome and then rejoin it. And nobody will understand because there is no marker associated with this technology. Could you get it?
Yeah, yes, sir. Yeah,
no marker. So people may not be able to detect it because this is totally marker-free business.
Yes, sir.
So you're absolutely right. People may not be able to detect it, and it can be considered as a mutant, wild type, or a variable. But that would be ethically wrong. That's why we discuss this in the ethics part. What should be done and what should not be done. Could you get it? But if somebody wants to do it, it will be very difficult to prove technically that this was engineered and what was not existing prior to this, right? So because there is no any marker associated with this, but in the transgenics you have markers which get embedded along with the gene of interest. So from there we can find out whether it is an antibiotic gene or some kind of a scoreable marker or a selective marker. So those becomes the evidence. But here there is nothing like that, as you have seen in any of these three technologies, right? Yeah. So I think I cannot give you the answer directly, but still I think you got a clear answer.
Sure, sir.
The concept is clear, right?
Yeah.
Okay, fine. Any other questions? So in the live session also, no any questions coming. Right. So how do you find out how many people join the live sessions? Where? From where? No, this is where? From where? But here, how can I find out?
First line.
Oh, 6 watching now. But we cannot know who are the people, right? So yeah. So if anybody in the live session has some questions, they can also ask. So these are two separate channels.
Yes.
But the live session people will be able to hear us?
Yes.
Okay.
Sir, can I ask general questions?
Yeah, sure. Please continue. Yeah.
So, sir, what do you think on CRISPR-Cas technology? How much years we are how much progress we have made as per USA and Europe and Australia with?
Oh, I mean in India?
Yeah. On CRISPR.
That's a good question. I mean, many people are now many research projects are going on in India. So and applications in various systems like plant systems has been also there for particularly for crop improvement. So, but anyway, in India till now, to the best of my knowledge, we do not have any CRISPR-Cas9 engineered product in the market. In the way we have in the USA, you have that mushroom, but in India, we don't have anything of that sort in the market till now. But I think some progress is being made, because here we are not developing any technology. We are only going for the application of the CRISPR-Cas9 technology in improvement of, say, crops or certain strains or commodities. So probably with some time, some of the products will be available.
So, sir, what will be the future of that product? Suppose if you are make any CRISPR-based chili, which is resistant to some disease, so what will be the future? Whether
so
the
that will
government
actually, yeah, that is what the government is trying to, provide a guideline, also a legal framework regarding this, taking the safety concerns. I mean, so all the and backed by scientific evidence. It's not like that it will be hypothetical. So in a way, earlier there were no any regulations, but now these also has to be brought under regulations, because ultimately it is a product, whether it is genetically engineered product or a genome edited product. So they have to meet the safety standards. So when people consume it, there is no any kind of harm. And second thing is also ethical. I mean, ethical in the way, and also biopiracy. Many times, somebody may take our strains and then get them little bit edited here and there. So thereby and later on claim that this particular variety or strain or species belong to their country, not our country. Could you get it? So,
yes, sir.
so there are many such issues. I mean, not just the product part, but also the ownership part, the biopiracy part.
Biopiracy plus patent.
Patent. So so I think by just tweaking some genomic stretch somewhere, somebody will later on try to claim, see, this is the strain species belonging to us because it has a very unique genetic sequence at this particular marker portion or in these microsatellites or whatever. They can engineer it, right? And that way we may lose precious bioresources. So there, it's a very I mean, important topic, not only just for producing, making products, but also for safeguarding the existing bioresources, which we are the rightful owner. Is this
yes, sir.
Clear to you, Rakesh?
Yes, sir.
Yeah. So you are working in the plant system?
Yes, sir.
Yeah, which university?
Sir, University of Agriculture Science, Dharwad.
Okay.
Dharwad, sir.
Yeah. So live session anymore?
Sir?
Yeah.
Sir, I want to ask about exam. I mean, the type of questions and pattern, type of questions which will be coming in the exam.
Priyanka, right?
Yes,
yeah.
sir.
So, yeah, look, I mean, the entire course is covered, all the 12 modules. And as per the NPTEL MOOCs guidelines, it is MCQ pattern, right?
Okay.
So that is as per, and I think you have also got the guidelines in the NPTEL MOOCs website. So
Yes,
it will be
sir.
as it will be as per the practice done by the organizers. So I think it clears you the doubt.
Yes, I mean, the questions will be similar to those which we have covered in assignment part.
Yeah, and then almost, and then but somewhere some calculations may be, although it will be MCQ, so it may involve some rough work separately to take a decision which is the right answer.
Yes, sir.
Right.
Okay, sir.
Yeah. So till now you have finished all the modules, right? Or
yeah, today is last date for 12th module.
12 module. Okay, fine.
Sir,
so you are liking the content of the course?
Yeah, sir. We like earlier means Crispr and means I was actually waiting for these courses, and I earlier last year I have also sent email to NPT course also. Means I was very excited to join this course, and I registered also. I'm doing good. I like also course. And I am another course I was seeing in molecular biology. They are maybe expecting maybe next semester they will launch that one course in our bioscience.
Molecular biology.
Yes, sir, molecular biology. That course was not there earlier.
Okay. So this was delayed due to various reasons, due to the COVID, because the recording, everything was disrupted.
Yes,
and
sir.
I also caught up COVID at a point of time, and my voice was totally gone for a couple of weeks. Yeah. But anyway, we could made it. Yeah.
Yes.
So certain things, a particular portion will be the practice on the YouTube channel, and then assignments, and also some majority will be, of course, new questions. So that is what I can tell you in brief. So I think it will be not difficult. The material is already with you. It will be only from the content that has been covered, not something beyond that.
Yes, sir.
Yeah.
Actually, I have, sir, other few courses I have given. So earlier means in that MCQs are there. Sometimes they will fill in the blanks. They will give us. Now latest last time when I have other course I have tried, they have just given fill in the blanks with direct no option was. So it was very tough. So
Okay.
for all, yeah. So for us,
But anyway,
time all
it is
yeah.
always from the content itself. It's not beyond the content, right?
Yes, yes, sir.
Yeah. Nice. So any other questions? Maybe Priyanka or Neela is there. If some of you would like to ask, let me first check if somebody is there in the YouTube channel and they have some queries. There is no any queries in the YouTube channel. So yeah, maybe if you have any queries, you can ask, and then after that we'll come to an end. Neela want to ask some questions? No. Priyanka want to ask something?
Am I audible?
Yeah, you are audible.
It's just a small clarification. iPSC models can be used to make vaccines more, or how do I say, tissue culturing for transplant organs also.
Can you repeat the question? iPSC can be used for as vaccines and
Vaccines, and can it be used for like tissue culturing, like recent technology has developed to making
okay.
organic models and stuff.
Okay, yeah, sure. See, I mean, that is very easily done. See, you are what you are trying to do is convert a somatic cell into a stem cell by
Yes.
by some kind of synthetic approach, right? We are not harvesting the stem cells from the organism. What we are doing, we are converting the somatic cells, body cells, into stem cells by some synthetic method. So once we convert them back into a stem cell, so they become totipotent, right? So now, once we have these totipotent cells through these induced process, so these cells are like the naturally found cells inside the body, naturally found stem cells. And now, depending on the type of cell we want to generate, we can differentiate them, okay, following certain protocols or processes, and further culture them, okay, and also use them in therapeutics. So you want to make liver cells, you can convert them into liver cells. You want to convert them into nerve cells, you can convert them into nerve tissue cells, or some skin cells, or any other lung cells, so on and so forth. So yes, it is possible to use them, induced pluripotent stem cells, in the tissue culture process.
Okay.
Right.
Thank you.
So I think we can stop for today. We are also about to complete 1 hour. So if you have still some queries, maybe you can mail us. We can get back to you through email. Right. So Rekha, we can wind up if you want right now. Yeah. So okay then, thank you all of you.
Thank you, sir.
Thank you, sir. Thank you, sir.
Yeah.
Thank you, sir. Thank you, sir.